It's my pleasure to welcome you to the Florence Mahoney lecture, which will be given today by Dr. Quiroz on the pathway to prevention of Alzheimer's disease based on her outstanding, groundbreaking work on studies of the Colombian kindred with autosomal dominant Alzheimer's disease. The Mahoney lectures are in memory of Florence Mahoney, who spent much of her life promoting NIH and including the foundation of the National Institute on Aging until her death at age 103. And up until that time, I would meet with her each year. She would want to know who was going to give the lecture and had high standards. She wanted no nonsense, wanted the most cutting edge, but also rigorous science. And she'd be thrilled to see how Yekil keeping up our tradition here today. So to introduce Yekil to you further, she began her career with her undergraduate education at in Antioquia, Medellin, where the kindred has become a focus of her study, has originated. She then went to Boston University for her both master's and PhD degree in clinical neuroscience, moved to Mass General, where she has now advanced to the level of associate professor in the departments of neurology and psychology. She has been honored by multiple, including the NIH Early Independence Award, one which encourages people with high promise early in their careers to move forward towards independence, and a great honor indeed. Other honors by the Alzheimer's Association and by MGH itself. And today we'll have an opportunity to hear from her about this groundbreaking work. I encourage you to have your questions ready for her, and we'll be back with them at the end of her talk. Yekil.
Thank you, everybody. Thank you, Dr. Hodes, for the kind introduction and also for the invitation to present our work. It's really an honor to give this lecture and be a fan of Florence Mahoney's work, especially her advocacy and leadership in NIA. So thank you again for the opportunity. Today I'm going to be sharing with you some of the work that we have been doing with the Colombian kindred with autosomal dominant Alzheimer's disease. And as it was mentioned before, so my name is Quiroz, and I'm right now an associate professor at the Harvard Medical School at Massachusetts General Hospital. And I wear several hats, so before I moved to the Colombian kindred, and based on some conversations that I have this morning, and thank you everybody that actually got some time to meet with me this morning, and also a big thank you to Kim Calvin for organizing all the logistics for this meeting. I appreciate it. So I'm also the director of the Multicultural Alzheimer's Prevention Program, and I'm not going to be mentioning this during the presentation, but I wanted to like I mentioned, that I'm very strong and very committed to increasing diversity and representation of women and other individuals from other underrepresented minoritized groups in research and science. And as director of the Multicultural Alzheimer's Prevention Program, we try to follow the cultural approach to dementia prevention. Just last month, we wrote this perspective review paper in Nature Reviews. So I invite you to, if you are interested in this topic, to actually take a look and pretty much learn more about the work that we are doing with diversity and increasing diversity in clinical care and research. With that said, so I'm just going to move to the topic for today. That's going to be based on the autosomal dominant Alzheimer's disease. So for the outline, the same, I'm going to start with an overview of what we know about autosomal dominant Alzheimer's disease. Then talking about some of the early evidence that we have with cognitively unimpaired mutation carriers. Moving to some of the work that we have done with the COLombia-BOSton study, especially what we had learned with the tau pathology in preclinical Alzheimer's disease. Then briefly discuss what we have learned about cognitive biomarkers, especially briefly mentioning the work that we have done in the past few years with COLBOS remote and the blood biomarkers. And lastly, I'm going to be discussing some of the work that we are doing currently with the COLBOS Extreme that is characterizing sort of the protective factors and cognitive resilience in the kindred. And just to make sure that everybody knows, I know that you're probably familiar with this, but just to make sure that everybody's on the same page, as a reminder, so the most common form of the Alzheimer's disease is the sporadic late onset Alzheimer's disease. This explains the majority of cases. Usually individuals that develop this form of the disease, they have symptoms over the age of 65. There are some important risk factors, older age being the most important one, but followed by family history, and also we know that the major risk factor, a genetic risk factor, is ApoE4. And there is also another type of Alzheimer's disease, that is what we call familial Alzheimer's disease, that is explained only a minority of cases, and is usually explained by autosomal dominant mutations in 3 main genes that are presenilin 1, presenilin 2, and the APP genes. Different from the sporadic late onset Alzheimer's disease, individuals usually develop Alzheimer's disease before the age of 65. These mutations are nearly 100% penetrant, and depending on the mutation, they have a specific age or clinical onset. In the past decades, and you also may be familiar with this, so we are moving to this definition of Alzheimer's disease as a continuum of Alzheimer's disease. And we actually know that the last stage is dementia. Let me see if this works. And it's preceded by the mild cognitive impairment. And the current treatments that are developed usually are given to individuals when they already have symptoms. And all the work that has been done in the past several years has been trying to identify individuals in this preclinical stage. And one of the goals of identifying these individuals is actually to be able to delay the clinical onset and, of course, in the future, be able to prevent Alzheimer's disease and dementia altogether. One of the goals of this prevention, or the preclinical detection of Alzheimer's disease, is to actually be able to initiate treatment as early as possible, so individuals have the best possible outcome. And together with the early initiation of treatments, we also have the possibility of being able to prevent Alzheimer's disease altogether in the future. One way that we have been using to actually detect Alzheimer's disease as early as possible is through biomarkers. And as you may know as well, so biomarkers give you the chance to detect Alzheimer's disease or any disease, also give you the chance to stage the disease, monitor the disease, and in clinical trials, they actually give you the chance to monitor the response to treatments. There are several biomarkers at different stages of validation, and here I'm just showing some of the biomarkers that we are currently pursuing at some point, including biomarkers using neuroimaging for brain atrophy, tau, amyloid, synapse loss, and also biomarkers that are more recently developed for neuroinflammation. And when you're talking about biomarkers, there are always 2 ways to go about biomarkers. One, you can study individuals that are at increased risk to develop sporadic Alzheimer's disease, or in this case, late onset Alzheimer's disease. And you can also go about studying individuals with familial Alzheimer's disease. And as you already know by the talk on my presentation, so this is the way that we have been working on these biomarkers. But this is something, as Dr. Rhodes mentioned at the beginning, that we have been doing for several years. As a matter of fact, was something that Dr. Francisco Lopera, the guy that is here in the picture, started doing like over more than 30 years in Colombia. And in Colombia, especially in Antioquia, we have families, large families with several neurodegenerative conditions, not just Alzheimer's disease, but also with frontotemporal dementia, Huntington, Parkinson's, CADASIL. And he has been characterizing these families for now over 30 years. And I joined his group when I was in college, so for me, a little over 20 years. And since then, I have been working with him in this project characterizing these families. So my main contribution to the work of the group in Colombia has been mostly focused on the neuroimaging and biomarkers, and of course, helping and getting the families ready for everything that has to do with clinical trials and treatments. So just to remind you a little bit, or telling you a little bit about these families that we follow. So the ones with Alzheimer's disease, we actually have the world's largest extended family with a single genetic mutation for early onset Alzheimer's disease. Right now, we have a little bit over 6,000 individuals that actually carry a single genetic mutation in the presenilin 1. This is the E288 mutation. We call it the PISA mutation. And individuals that carry this mutation, they actually develop mild cognitive impairment about the age of 44 and dementia about the age of 49. And the same, there are several similarities in clinical cognitive and biomarkers between this form of the disease and the most common form of the disease that is the late onset sporadic AD. And of course, there are also differences between the 2 types. But having the opportunity to study these families also give you the opportunity to study individuals that are pretty much destined to develop Alzheimer's disease dementia in the future, but study them several years or even decades before they have any symptoms. So it has been a window of opportunity to characterize some of the preclinical changes related to Alzheimer's disease. And that's exactly one of the first questions that I have been very interested in answering, and I'm still kind of like working on that. It's like, what are the earliest biological and physiological changes that are actually associated with the predisposition to Alzheimer's disease? And to start answering that question, the first set of the studies that we did was actually using a functional MRI. This is some of the work that I did as a PhD student. I actually used task-related fMRI to study brain activation when the carriers and the age-matched non-carriers, that in this case are pretty much the siblings, were actually learning face names when they were in the scanner. And the first study was this one, that it was older carriers, individuals older than 25. When we compared the learning during the novel face names compared to the repeated face names, we found that the carriers, they actually have hippocampal hyperactivation in the anterior hippocampus compared to the non-carriers, which was already telling us that the carriers have some brain abnormalities in brain function, or in this case, learning face names. Then I decided to go younger, so we went to the 18 to 25, and we did the same task. They were using the same face name task. And then we actually saw that in addition to this hippocampal hyperactivation, they also have hyperactivation on other regions, including the parahippocampal gyrons and other regions of the brain. So pretty much there was still some abnormalities in the carriers compared to the non-carriers. And then I went even younger, and I included some children, in this case individuals from 9 years to 18 years. And then the first thing that we found, it was that there were no difference in the middle temporal lobe between carriers and non-carriers. And when we look at the parietal regions, we saw this hyperactivation in the carriers compared to the non-carriers. We call here less deactivation of parietal regions, that pretty much because we don't expect this activation during this task. So these three first experiments were pretty much telling us that there were some abnormalities in the carriers several years before they have any kind of symptoms. Then I started looking at the structure, and the next experiment was just looking at cortical thickness, and the same looking at the older carriers. We actually found that the carriers compared to the non-carriers, they actually have thinner cortex, and especially in the parietal regions, a few years before symptoms. And then I used ERP, that is even related potential, so EEG, based on EEG in this case, and the carriers also have abnormalities in posterior regions when they were learning, during learning, compared to the non-carriers. So these 5 experiments altogether provided some of the first evidence for brain changes in cognitively presymptomatic individuals many years before clinical onset. After all these studies, so we started collaborating with the Banner Alzheimer's Institute, and then we used other biomarkers, including biomarkers in CSF. And the first study that we did for amyloid PET, and putting all the biomarkers, these biomarkers together in this age-related trajectory, we actually observed that the first abnormalities in these individuals that are pretty much the same individuals that I showed before, the earliest abnormalities that were seen were changes in amyloid beta as measured in CSF, followed by changes in amyloid as measured in PET, as early as in the early 20s. So one of the first questions, and the same, like you will see that in this presentation, I have actually more questions than answered. One of the first questions that I have as a result of this is pretty much the same. Like now that we have evidence of these abnormalities, so we know that there are abnormalities in brain function, but like what are actually the earliest abnormalities? And that actually brought me to another couple of questions that I still don't have the answer. And it's pretty much like, are there any neurodevelopmental differences between the carriers and the non-carriers? And in case that there are neurodevelopmental changes due to this mutation, are these changes actually contributing to future cognitive decline in the carriers? And the same, like to start answering those questions, which I still don't have the answer, I want to bring your attention back to the first study that I showed you with the children, when in addition to the abnormal activity during task-related fMRI, We also found abnormalities in resting state network, as seen here. We found increased connectivity between the posterior regions and the middle temporal regions. And we also found greater volume, greater gray matter volume in parietal and temporal regions in the carriers compared to the non-carriers. Pretty much like even at 9 years old, that is the youngest ones that we have, we are seeing these neurodevelopmental changes in the carriers compared to the non-carriers. So I'm still very intrigued by this, but like having all the imaging that we have with these individuals. So together with one of my PhD students, we actually put all this data together, and we start looking at the lifespan. And this is one of the first attempts to look at that data all together. And we put together over 200 MRIs that we had. That's pretty much everybody that I showed before. And this is kind of like what we found that is very interesting. We found that the thickness, for instance, that is the first measure that we included here, is actually fluctuating across the lifespan. And when we actually ran this model, we found two cut points that pretty much one that it was very clear here at the age of 17, and another one that I was seeing here at the age of 32, when they clearly start declining until, of course, they start developing MCI and dementia. So I was still very intrigued about what's going on here before the age of 17. So we went, and the same together with my PhD student and another PhD student, Araville, we went into the cognition, and we collected data, in this case in over 1,000 children from the Kindred, using this measure, that is the WITS-4, one of the first measures that were available. And we didn't find difference between the carriers and the non-carriers, but when we start taking a closer look at the data, we actually found sex differences in cognitive abilities between the children in the way that the boys with the mutation, they have decreased working memory compared to the girls with the mutation, and also compared to the ones without the mutation, which pretty much suggests a sex-specific genetic risk in this early life cognitive performance, which is the same. Like for me, it's very interesting, and it's pretty much telling us that there is something there in neurodevelopment that needs to be further explored. And even more than that, like that this increased risk of future cognitive abilities, especially the ones that we are seeing in the boys, may have important implications for these children, even when we are thinking about learning and academic achievement, and even in the future, may be related with the sex difference that are seen in memory measures, or then we are thinking in difference that we are seeing in resilience and pathology, for instance, and later in life. So the same, this is to be continued. We don't have the answers, but I wanted to share this because I think that it's very interesting. The other question that came out after all the initial work is the tau pathology. We didn't have tau pathology. Unfortunately, in Colombia, we didn't have that technology. So it was something that I was missing by the time, like the same, we have CSF, we have CSF and we have a MRI, but we didn't have a PET. So for this lesson, I thought that it was very important to actually have that piece of the puzzle to understand how the tau pathology in relation to amyloid pathology fit in the trajectory of the disease. So in 2014, and Dr. Rhodes mentioned this at the beginning, so actually apply and receive one of the early independence award, one of the DP5 grants, and as part of the DP5, actually proposed to bring the Colombians to Boston as part of what I call, the nickname is COLBOS, that is the Colombian-BOSton study. That is pretty much bringing to Boston to have the neuroimaging that was not available in Colombia, and trying to answer that question, what happened with tau pathology in the brain and how it fits in the picture of the other biomarkers. So what we have learned from the COLBOS, that right now we have 81 individuals that actually came for baseline examinations. So what we published in the first report is that the carriers, they actually have amyloid pathology, what we call amyloid positivity, as early as they are 28 years old. We also learned that they have increased tau pathology compared to the non-carriers in their late 30s, Now we know that it's as early as they are 36, few years before the clinical onset. We also now know that the accumulation of tau pathology begins in the entorhinal cortex and spreads from the entorhinal cortex to other regions. We also learned that increased levels of tau were only seen in the setting of substantial cortical amyloid deposition. All the individuals that actually have high levels of tau were those that actually have already really high levels of or amyloid. And we also learned that the tau pathology was highly correlated with measures of amyloid deposition and with cognitive decline. And now we also know that amyloid is better correlated with cognitive decline than amyloid. The other thing that we learned here in the pictures, as you can see, is like once they start developing mild cognitive impairment, tau pathology is spread in the neocortex, so is widely spread. Just recently, and this is something that my colleagues, Dr. Guzman Velez and Dr. Diaz, were leading this effort. So we start like at the same, going back to the connectivity, which I'm still very into connectivity. So we look at we try to examine the association with tau pathology and brain connectivity, and using the approach of using voxel-based approach, see if there was any association with tau pathology and the functional segregation and integration in the brain networks. And we actually found that the mutation carriers actually had less functional segregation and integration in the posterior default mode networks, as you can see here. And very interestingly, we also, when we took out the association with tau, we found that the greater tau pathology was actually associated with lower segregation and integrated functional connectivity of these networks. So pretty much here in this first report, we actually were able to show that even at those levels of tau pathology, which in the COLBOS sample, they are very incipient levels of tau pathology, so they are still just starting the accumulation of tau pathology in their brains. Those levels are already impacting the brain functional organization several years before they had cognitive impairment. All of this, the same, like I didn't show the descriptions, but most of the individuals in COLBOS, they are cognitively unimpaired. We only have 7 that are with mild cognitive impairment. In 2017, we started with COLBOS-2, that is the longitudinal biomarker study. And as part of the longitudinal biomarker study, we invited individuals to come back for follow-up examinations. And as part of this study, they were coming back every 18 to 24 months. And this part was running very smoothly until COVID. But, so I'm going to show you the ones that we collected before COVID. And so far, we have 35 that have been able to come back for the three times. So as part of the longitudinal follow-up, this is something that Justin Sanchez was leading. So we found that carriers, compared to the non-carriers, they actually had greater tau pathology and amyloid as predicted. As you can see here, the distribution is very standard. Something that was very interesting, and I still I found very interesting, and it's something that with more subjects that we have been able to confirm. Here we have the carriers and impaired carriers that are in blue, the few that we have there for impaired in red, and then the controls. You can actually see that the unimpaired, the accumulation of amyloid appears to be a steady process, beginning as early as in the 20 years prior to dementia onset. And here is amyloid, and this is age. And in B, you can see tau, and here I'm showing tau in the precuneus compared to age. You can see that in contrast, tau pathology is very rapid and is widespread in the neocortical tau, and you can see that it actually, nothing much happens in their early 30s, but as soon as they get closer, you can see this rapid increase of tau. And the same, now that we have more subjects, the pattern is very similar as well. So tau and amyloid changes, the measures are very correlated. Tau baseline and change are also very linked to memory decline, as well, and more linked to memory decline than amyloid. With all this data, as you can see in COLBOS, we have trying to many different sub-studies with different research questions, but all of them using the biomarkers and trying to improve biomarkers, and also understanding protective and risk factors for age-related cognitive decline and dementia, using this model of autosomal dominant Alzheimer's disease as the framework. We are very interested in understanding sex differences in this Kindred, for instance, also understanding the role of sleep, physical activity, chronic stress. We are now also testing other biomarkers, eye biomarkers, understanding the role of cardiovascular risk factors, and of course, several other factors as well. And I don't have time to go out through all the studies, but I'm just going to highlight a few of them just for you to give a sense of what we do. So I'm going to start with some of the applications of the work that we have been doing, starting with early diagnosis of cognitive decline and dementia. And as you can imagine, everything that we do with the Colombian Kindred, we are doing in Spanish. So we have taken this opportunity to actually validate and develop some of the measures for testing Spanish speakers, and this is one example. So the face name associative memory exam, face name, was one that it was actually already showing to be sensitive to preclinical Alzheimer's disease. We actually created the Latin American version, validated that version first in a Colombian sample, created norms for clinical use, that is this paper. And after we created norms for clinical use, we actually used it in COLBOS to test it in cognitively intact individuals to see if the measure was actually able to detect carriers from non-carriers, and also if it was correlated or not with tau pathology. This is some of the work that one of my colleagues, Dr. Clara Villa-Castelar, was actually leading. This is how the task looks. The task is actually available as part of the NIH toolbox. So we have the learning, then they learn the face, and they had to identify and recognize the face, the letter, the initial, and the name. So what we actually found with the COLBOS data, this is including only the ones that are considered cognitively intact. That means that they don't even have concerns of memory, like as clean as it goes. And we actually were able to use this task to show that only the performance in the task was able to distinguish carriers from non-carriers, and you can see here. Just the immediate recall and also the total recall, identifying the carriers from non-carriers, even within the normal range of performance. We then look at the correlations with pathology, and here I'm showing the performance and amyloid pathology, and the worst performance in the test was actually correlated with greater levels of amyloid, the same in individuals with no cognitive concerns or cognitive decline. And then we did the same with entorhinal, and worse performance was associated with greater levels of pathology as well. Then as exploratory whole brain analysis, we used the associations with performance and the tau pathology here and amyloid here, and you can see that the regions of the brain that were more associated with performance were actually the temporal lobe and the parietal regions, and the tau in particular, that are the ones that we know that are very sensitive to early accumulation of pathology as well. Once again, in individuals with no cognitive symptoms. So it's a promising task, and we continue to use it as a longitudinal, in the longitudinal study as well. With COVID, as many of you probably also have to adapt it to the remote testing, we did too. So I actually created the COLBOS remote, and it was pretty much a sub-study that it was in Colombia. When we recruited, we created a cohort of individuals that pretty much were that underwent remote testing, blood sampling, and all the remote testing was actually done either in the computer or in their smartphone. So we used that opportunity to actually validate some of the measures that were designing for remote testing. And we have so far 4 measures that are novel measures that are at different stages or piloting. And here I'm just going to share one that is actually the one that is more advanced. All the measures that we have in different steps, they are multicultural, multilingual. So we have right now in English, Spanish, Korean, and we are now developing one in Portuguese as well. So the idea is to have this available in the future for anybody that wants to have access to these measures. So the one that I wanted to share with you as an example is the memory for semantically related objects, or the mesero. And here is one that the same, like my PhD student was the one leading this effort. We have objects that are semantically related versus objects that were semantically unrelated. And they had to learn this. This is based on the delay to match sample object test, and they had to just say if the object match or no match after a few seconds delay. And what we found is saying, like, this is including individuals, only the individuals that actually completed the task in the computer. So they were given the option of completing in the computer or in the smartphone, but here for this analysis, we only include the ones that completed in the computer. So we have 35 that were carriers, and 26 that were non-carriers, and 5 that were MCI. And what we are seeing with the performance is like the total performance was able to distinguish carriers from non-carriers. It was different even within the normal range. And also between the two conditions, the related trials were actually the ones that were contributing to that difference between the carriers and the non-carriers. When we look at the other two conditions that were 4 items versus 6 items, only the 4 items, for instance, was the one that was distinguishing the carriers. And this is comparing the unimpaired versus the non-carriers. Of course, the MCI, they performed lower in everything. But the same, like, this is just an example to show you that the opportunity for designing measures that can actually detect or be more sensitive for preclinical and subtle changes in cognition to detect or identify individuals that are going to be developing cognitive decline in the future, instead of using the same measures that we have traditionally used to identify individuals with mild cognitive impairment or dementia. So the other component that I wanted to mention here is the same, and this is part of this early detection. As many in the field, so we also have taken a look at blood biomarkers, and this is something that in the past, like Dr. Lopera has collected blood samples in several individuals. So we started looking at the data that was already collected in the Kindred. So in the first 2 papers that we have, that we published like 2 years ago now, we look at levels of NFL, that is one of the markers of neurodegeneration, and also the p-tau 217, that is one of the markers of p-tau. And in the first paper, we actually included about 2,000 individuals, 2,000 samples from the Kindred, and we showed that the marker was actually able to distinguish carriers from non-carriers above the age of 20, and the same was true for the p-tau. So they distinguish carriers from non-carriers, like in the age of 20. But here I'm showing you when we actually put together the carriers from the non-carriers, it seems like the p-tau was a little bit more sensitive for the earlier changes than the NFL. So distinguished a little bit earlier than the non-carriers. But the same, like having this opportunity to have this data from biomarkers, I think that it opens also the window for a lot of opportunities in terms of increasing access, for instance, to individuals that don't necessarily have access to, for instance, in this case, to get tau PET imaging or amyloid PET imaging and so far. So one of the other questions that we have here, and this is something that we're still like a work in progress, because little is known about this association between, for instance, markers of plasma or PET measures of tau, and especially when we're talking about longer intervals, right? It's something that we can predict, use one to predict changes in the other one when it's like a longer interval of time. So since we have some data from tau PET pathology in COLBOS, we actually use the ones that had already pathology, markers of pathology in COLBOS, and look at the ones that actually had these broad samples in p-tau in this paper that we just showed you, and ask the question is if plasma p-tau could actually predict subsequent levels of tau PET pathology and cognitive performance. And I'm going to explain a little bit more in detail what we did. So these individuals, they actually have plasma tau in the site that I just showed you. And then about 8 years later, they came to COLBOS. And in COLBOS, they have tau PET, amyloid PET, and of course the measures of cognition. So we use this opportunity to test if these levels of plasma that they get at what we call baseline would actually help us to predict how they will look 8 years later. And of course, the sample size is small because we don't have a lot of people in COLBOS, but we had 24 that were carriers and 20 that were non-carriers. And so this is pretty much the study that we're doing now. And look at only with 24 carriers how beautiful this looks. So first, we were able to replicate that carriers actually had higher levels or p-tau 17 compared to the non-carriers, and also that the plasma p-tau is positively correlated with older age. But then, the same like when we actually look at cognition, and here I'm only showing one test, but it's pretty much across all the different cognitive measures. Like actually p-tau predicts worse cognition across all the different cognitive battery that we have, almost 8 years later. And then the same, like when we actually look at pathology, we had plasma p-tau, and then it's associated with greater PET pathology in amyloid and also in tau. And here I'm only showing entorhinal because, as I mentioned, it's the first one that we see changes, but it's not only associated with levels of entorhinal, we can also see that it's associated with inferior temporal and precuneus, and the same like in the old carriers, even only in the unimpaired carriers. The same, almost 8 years later, this is still predicting these measures. We run some exploratory analysis, and this is with all the carriers. You can see that especially with tau PET, the temporal regions and the parietal regions seems to be the ones that are highly correlated, and this is with all the corrections that we use. And even with these corrections, where we actually take a look at only the unimpaired carriers, the temporal lobe regions and the precuneus are still there, almost 8 years after. So I think that this is, I'm biased, but I think that this is very beautiful. So baseline levels of plasma p-tau actually predict subsequent levels of amyloid and tau burden, and of course also future memory performance in the carriers. And these results, of course, they are very preliminary because the sample size is not that big, that large, but they provide some support for plasma p-tau as a potential diagnostic and prognostic marker or AD pathology and cognition, and with some promising utility in clinical practice and trials. As I was saying, like as a possibility of having blood biomarkers to increase access to early detection of Alzheimer's disease, or at least to detect who are the ones that are at increased risk to develop Alzheimer's disease. And the last thing that I wanted to mention is the project that we had that is COLBOS Extreme. For the lack of a better name, so I call it Extreme because for this one, I just wanted to go to the stream. That is pretty much to go after the ones that actually remain unimpaired as part of the Kindred to see if we could find some clues for protection against cognitive decline and dementia. For this one, the same, like it was launched in 2017. And as a reminder, individuals from this Kindred, they usually develop cognitive impairment around the age of 44 and dementia around the age of 49. So we were after the ones that were older than 60 or 65. So the first case is the case of Doña Lidia. That is the one that we reported in 2019. And Doña Lidia, she was 72 when she started showing signs of cognitive decline, almost 3 decades after the expected age of clinical onset. And the same, her family brought her to Dr. Lopera. Oh, sorry. Her family brought her to Dr. Lopera in Colombia. And by the time that she was seen by him, so she was relatively healthy, and the only thing that was relevant in her medical history was that she had really high levels of cholesterol, but nothing else was significant. So she was enrolled in COLBOS, and as part of COLBOS, she came to MGH for imaging, and I'm going to walk you through the imaging findings here. So, can I? Oh, no. Oh, look at that. You are not seeing what I'm seeing. Okay, so here you can see the amyloid pathology, and all this time I was just clicking here, and I thought that you were actually seeing what I saw. So, I apologize for that. So the amyloid here, this is her compared to the typical MCI. And you can see that her levels of amyloid were really high. Actually, the highest levels of amyloid that we have seen in anybody in the Kindred. And the second row here is the levels of tau. So this is her compared to the typical MCI. And you can see that her levels of tau pathology were very limited, and not just very limited in terms of the amount, but also in the location where the tau was actually accumulating. And the other way to see it is here. So she's the one in red, and everybody else is everybody that we have at that point in the Kindred. The black one, I missed it there. The black ones being the MCI, and the gray ones being the non-carriers. And the same, like you have amyloid, here, and you have tau here. She's also the one in red. So high levels of amyloid, very limited levels of tau. We also ran some structural imaging on her, and the same, she's the one in red. So her levels of atrophy were very limited when we see him first. And we also did some FDG PET, and she had no levels of glucose metabolism when we saw her. That's pretty much what we see there, and also what you see in this picture right here. So the imaging showed that she had very limited tau pathology and neurodegeneration, despite having really high levels of amyloid. The other thing that we did is that we took a really close look into the tau levels, and this is like a flat map of her temporal lobe cortex. And the same, when you see the ERC, that's the entorhinal cortex, and you can see that she has a little tau in the entorhinal cortex, but nothing compared when you actually look at the C and the D panels. So the C is somebody that is asymptomatic from the Kindred, that is a 38-year-old from the Kindred, and when you compare her with a control, that is the same control that we have in the previous slide, that it was a 44-year-old carrier. So pretty much her levels of tau were extremely limited compared to anybody in the Kindred. So we ran some genetic analysis on her, and we actually found that first of all, we confirmed that she was a carrier of the PISA mutation, the E288 mutation. And we also found that she was homozygous for the rare mutation, that is APOE3 Christchurch. So to understand the potential mechanisms of this mutation, we decided to look at the location of the mutation. And in this region around the amino acid 136, as you may know, is critical for two functions. One, it binds to the LDL receptor, and also it's important for these interactions with the type of sugar that are the glycosaminoglycans, especially the heparin and the HSPGs. So previous work by others has shown, and you probably are familiar with this as well, that the LDL receptor has a higher affinity with ApoE4, followed by ApoE3, and then followed by Christchurch. The same, like this is something that we already know, like Christchurch actually has an affinity about 40% binding to these LDL receptors, but something that was not known by then, it was like the role of Christchurch, for instance, in this binding with HSPG. And that was something that, based on the location, as I mentioned, it was kind of like predicted, but it was not tested, or at least it was not reported in the literature. And for us, it was very clear and relevant at that point that going after HSPG was kind of like important, because as you can see here, HSPG has been implicated in several steps of the pathobiology of Alzheimer's disease, including the regulation of base activity, aggregation of uptake, or amyloid aggregation and uptake, amyloid-induced neuroinflammation, and also neuronal tau uptake and propagation. And it was kind of like interesting for us to see, like, maybe for some kind of this protection like Doña Lidia had, that it was 30 years for somebody like in the Kindred. Maybe for something like that, the mechanisms of the protection needed to be something that actually crossed the pathobiology of Alzheimer's disease at different levels, all the way from amyloid, neuroinflammation, tau pathology, and it seems like maybe something like HSPG could actually provide that answer. So then I pass it to my colleagues, the basic scientists, and some of the experiments that were conducted here, it was actually conducted at Dr. Arboleda Velázquez's lab and his postdocs. And one of the first experiments that they did was to start understanding the molecular consequences or Christchurch. And the first one, they actually measured amyloid fibrillation in vitro, and they actually found that Christchurch was compared to the ApoE3 wild type, actually have lower aggregation. you can see here. In the second set of experiments that they conducted, they actually used a heparin affinity chromatography just to start understanding these interactions between Christchurch and HSPG. And they found, as we can see here, that the interaction, for instance, the strength of the interaction with ApoE4 and heparin was very strong. It was less strong with, I am still pointing here, sorry. Okay, it was very strong with ApoE4, less strong with ApoE2. And what it was very remarkable, it was that the association with ApoE3 Christchurch was much weaker compared to the other two. That is something that you can see here. Quite remarkable, the weakest strength of association that was observed was actually the one with Christchurch. And then, as a proof of principle, we actually designed and to test whether these mechanisms could actually translate it into therapeutics in the future, we designed an antibody to actually mimic the role of Christchurch around this location of amino acid 136, and this is what is presented here. Pretty much you can see, you can actually see that when you actually use the antibody that actually recognizes ApoE, the antibody that actually recognizes ApoE3 around the amino acid 136 actually mimics the role in inhibiting the binding to heparin. So pretty much all together, the conclusion of these three sets of experiments is that first, Christchurch in vitro, all of these three are in vitro, actually regulates and modulates the aggregation of amyloid, which is intriguing in itself, especially the fibrillation, since, as I mentioned before, she had a lot of amyloid on her brain, so perhaps it could have been even worse if there was not for Christchurch there. Then we also found that Christchurch impairs this binding to heparin, and also we also showed that an antibody that actually recognized ApoE around this region of amino acid 136, when Christchurch is located, could actually inhibit the binding to heparin and suggest a potential therapeutic right there. And the same is something that, based on these findings, my collaborators and several others in the field, I know that are currently pursuing this as a potential therapeutic. So the main conclusion of this case is like the same. We learned that the impact of ApoE is not limited to amyloid aggregation, but it's also something that may impact, or directly or indirect, the downstream effects, especially tau propagation. We learned that this protection, especially in Doña Liria's case, may be mediated, at least in part, by the magnitude of this binding between ApoE and HSPG. And we also learned that maybe antibodies, something else like a small molecule gene therapies, that may interfere in this binding between ApoE and HSPG, or something that can reduce HSPG functions, may actually have a major impact on the treatment and prevention of Alzheimer's disease. So we have been busy working on this for the past few years, and hopefully in the future I can share more about these updates. But the same, like some of my collaborators are working, of course, in understanding the mechanisms of this neuroprotection, running more in vitro and in vivo models. We are also working, and I'm going to briefly mention some of the work that we are doing, understanding the impact of Christchurch in heterozygotes when we only have one copy. And as I mentioned before, so Doña Liria is only the first one. In the project with COLBOS Extreme, we actually were able to identify other escapees or other outliers, and we are in the process of characterizing the other ones as well. So as a quick update, as an extra pretty much, so with Dr. Lopera and others, we are trying to answer if ApoE Christchurch can be protective in heterozygotes. So in collaboration with Dr. Eric Reichman and others in the Banner Alzheimer's Institute, we actually characterize ApoE, we're able to characterize ApoE in 2,000 individuals from the carriers, from the Colombian carriers, including the Christchurch mutation. And we actually identified 97 of the Kindred members that actually have the Christchurch mutation, including 84 who are living and 13 who are deceased, but out of those 13, 7 who are actually brain donors. Out of these 84, 29 of them, they actually have both mutations, the ApoE3 and the presenilin-1 E288 mutation. And as you can imagine, so we are following this 84, and we start characterizing these individuals in the past year. So in the past year, all the 84 were recontacted, and they start undergoing comprehensive clinical assessments, MRIs, and FDG PET, pretty much all the imaging that it can be done in Colombia. So, sorry. Did I do something wrong? The age range for the carriers goes all the way from 17 to 72 years, and includes 11 that have the 2 mutations that actually are from 50 to 72. And from those, pretty much as I mentioned before, so 50 is already older than the typical median age or dementia onset. And the same, like we only start characterizing them for AIC, so we send this as an astra. So for characterizing the first 3, the cognitive performance that we review for these individuals, it actually fell within the normal range compared to the age-matched individuals from the Kindred. And to date, the same that we have 18 that has completed MRI and FDG PET. So hopefully by the time that we presented in AIC, we will have data on those individuals as well. So we had one individual that actually have traveled to Boston for tau imaging. And going back to the flat map of the temporal cortex, this is how the tau imaging in the temporal cortex look for somebody with only 1 copy. So the same, like compared to anybody in the Kindred, even after 2 years of follow-up, the levels of tau pathology, they are relatively limited. So the main conclusions that we have there, this is very preliminary, but first of all, the prevalence rate of Christchurch heterozygosity in the Colombian Kindred is very high compared to the general population. That's something, the first big news there. The preliminary data that we have is very preliminary, but suggests that they may have some level of protection, which we still need to further study. And as you may imagine, so we need to do more longitudinal studies and continue with the characterization, but the idea is to see if Christchurch with only 1 copy can be protected, or it's only protected for the ones in the presenilin-1 mutation. So all of these, like the same, so how much we can actually learn from 1 patient, and this is going back to like the clinic, like, you know, it's important to take it from the clinic and how much 1 patient can teach us about genomics, basic science, and hopefully in the future also teach us about drug discovery and clinical trials. And to conclude, so hopefully I convince you that the work with the families with autosomal dominant mutations provide a unique opportunity to study disease progression from preclinical to clinical stages, to develop and improve measures for early detection, to develop and implement novel interventions and clinical trials. Future longitudinal studies of these populations is needed to further characterize the biomarker trajectory. There is way more to learn from escapees of the autosomal dominant mutations, those individuals that are unimpaired, and of course, so we should include more families in the future. So as you imagine, this takes a village, and this is not an exception, so I would like to acknowledge everybody that has been part of the COLBOS study, everybody in my lab, my mentors in Harvard Region Brain Study, Dr. Risa Sperling, and Dr. Keith Johnson, Dr. Francisco Lopera and the Group of Neurosciences, Dr. Eddie Ryman, and all other my collaborators, and of course, the families in Colombia for their dedication to research and my funding support as well. Thank you very much.
I did. Would you like to start with a question? I have a few here. First, let me repeat the CME code, and that's 37950. I'll ask one question then, Dr. Hodes. I'll try to read this slowly. Heparin sulfate protoglycans are binding sites for multiple human viruses. Have you looked for signs of virus infection as a possible risk or protective factor?
Really good point. The answer is no, we haven't, but we should. Yeah.
Okay, thank
Thank
you.
you. Yeah.
Yeah, Gil, a couple of questions. First, among the many striking findings, one that was a surprise you mentioned, if I heard it correctly, is that carriers had an increase in cingulate cortex gray matter. Do you have any idea what makes up that increased mass?
So no, we don't have a design, like one of the things that we discuss in the paper, and this is like going back to the, I think you probably refers to the findings in the lifespan, the children that we got. So one of the things that we discussed that it could be like something related with inflammation.
Yeah.
But yeah, we haven't tested directly. It's just like one of the hypotheses.
And it's early, so you don't have necessarily post-mortem examination yet to...
Exactly.
Another very different kind of question. You know, in the Diane study, which I know you'll know well, it's another dominantly inherited Alzheimer's network that looked at a much smaller individual families and multiple of them across the world. I just know the behavior, the sociology of those families is that often they don't want to know their typing, and that leads to interesting strategies for clinical studies and trials. Can you comment on whether there's any such phenomenon in the Colombian cohort?
You mean if they don't want to know their status?
Yes.
Okay, this is a very good question, and it's something like the same. Like right now, for instance, and we are going to present some of the results of the pilot study that was conducted about that at AIC. So you pretty much, I'm not going to tell you everything. It's going to be there, but it really depends of like nuclear family by nuclear family. So there are some families that they are more inclined to know, and some other families that they really don't want to know. So like, yeah, so it varies.
Yes.
My turn again? Okay. This is from one of our researchers here, Paul Parcon. Very beautiful results, exclamation point. With regard to biomarkers, a lot of the data and testing presented related directly to amyloid and tau. What is the data available related to the inflammatory aspects of the disease in this kindred? Are there blood or PET biomarkers that were done? This may be especially important in the APOE carriers as well.
Yeah, this is an excellent question. The answer is like we actually haven't measured markers of the neuroinflammation yet, but as I mentioned in one of the slides, it's something that is ongoing. So I mean, like we are now actually collecting, especially CSF, and we are going to start measuring markers of neuroinflammation in CSF, and of course, in the future, we also hope to measure in plasma, but markers of neuroinflammation is better to measure in CSF. So we are collecting in CSF. In the future, we will have an answer for that, but we haven't actually directly measured it.
Please.
Hi, thank you very much. That was a beautiful presentation. In regard to the PET imaging on the Colombia kindred, can you tell if they had a similar heterogeneity to what has been described in sporadic Alzheimer's disease? That is, some that are more like posterior, other more anterior, other hippocampal sparing, or do they look by PET tau imaging very similar? I mean, pretty homogeneous.
So I can give you like two answers. So the first one is like, so the sample that we have in COLBOS is very small and is very homogeneous. So pretty much like, for instance, something that we have seen is because we started like, for instance, measuring tau in other mutations. Like the same when we start looking at other mutations, the pattern of tau accumulation starts being different. So the same, one of the things that we have with the PISA mutation or the E288, like tends to be like a very homogeneous. So that's one thing. So when you actually look at the pattern that we have, it's very, the pattern of accumulation of tau is very typical or what is called like amnestic, what you expect to see in an amnestic Alzheimer's disease. But the same, like as soon as you start introducing other mutations, you start seeing like, the heterogeneity of the patterns.
Thank you.
This one's kind of interesting because I was wondering the same. I've seen studies back and forth saying that coffee consumption raises the risk of Alzheimer's or reduces the risk.
Yeah.
This is great. This data set being from Colombia, have you examined coffee consumption in Alzheimer's?
So we haven't. So yeah, so we haven't, we haven't actually, well, let me see.
I don't know if that's an assumption that coffee consumption is higher in Colombia. I don't know if we
No,
should
the
make
point
that.
is that I
That's where it
think
comes
that-
from, but
Oh, I think that everybody drinks coffee, so at the end of the day, like, I will have like, so we actually measure like eating habits, for instance. So like we probably can take a look and see if there is any difference, particularly if how many they drink coffee and stuff like that. So as part of COLBOS, we are also measuring like lifestyle and stuff like that. And we have a questionnaire asking for eating habits. So and we ask for like what they have, or what they drink, so we could actually look at that specifically, but yeah, we haven't analyzed that data yet. But like everybody actually has a coffee in the morning, so it's kind of like even carriers, non-carriers. So I think that, but we can actually look at the data we have.
I have one more quick one from this. Thank you for the fascinating talk. Perhaps I missed this. How long has the Colombian kindred been known? And is this based in indigenous populations or European/Spanish populations?
So the mutation in Colombia was discovered in the late 80s.
Oh, recent.
So, and the origin of the mutation was actually tracked back to a conquistador from Spain. So it was tracked to Spain.
Yeah, I was just going to ask the more general form of the question about coffee, and you answered it already. In addition to looking at genetic risk or protective factors, in this population with a single homogeneous mutation, another great place to be looking for other non-genetic factors, and you already answered, I think, that, so you're looking at various other environmental exposures that might be protective or
Correct.
aggravate or accelerate.
Exactly. So yeah, we collect all the data, and it's, yeah. So we're looking into that and see if that plays a role.
So has the Colombian cohort, have they donated brains?
Yeah.
Yeah. So, and are you working on studying the profiles at that stage of?
Yeah.
Yes. Okay. Any interesting revelations there?
You mean for the case of the Christchurch? Or
Yeah.
or everybody?
Yeah. Actually, that would be great. Yeah.
Exactly. For the case of the Christchurch, we are currently working on the postmortem. Listen, I didn't mention, but unfortunately the patient died a little bit over a year ago, and the family actually donated the brain for analysis. So we are currently working on the analysis. And the same, like in COLBOS, as you saw, most of them are preclinical, so they are still very young. But the same, as part of the work that we do with the families, we always talk to them about brain donations. And in Colombia, they actually have a brain bank, that has over 300 brains from the mutation. So yes, so we will actually be looking into that. And the same, every time we talk about the importance of donating the brains.
Terrific. One other question, just for my own knowledge. So you had a figure where you were looking, you have different profiles for amyloid pathology and then tau, levels of tau, and you saw a gradual increase from 28 years in amyloid, I believe, and then this really sharp spike in tau at 30 years. So what's your hypothesis of what's going on in this age-related milestone that's causing that sharp increase in tau?
So the definitive answer is I don't know. I think that definitely there is something there that we are trying to identify what it is. So some of the hypotheses it could be neuroinflammation that is there in between, but the same something else that I didn't mention because it's something else that we are exploring. It could be also role for white matter, for instance. That is something that we are currently examining as well. But probably there is another factor there that is also contributing.
Right. Thank you.
Any concluding remarks, Dr. Hodes?
Yes, just want to thank you so much for a fascinating talk, great original work, and maybe an exemplar for everyone to be inspired how early in career one can find a place, a question for novel research areas, and really accelerate progress the way you have. It's been an outstanding story and a privilege for NIH to play a role in supporting it. We apologize for the intensity of the meetings you had this morning, and now you can rest and relax. And I think we've got about enough time to let you see the Florence Mahoney Memorial Garden in time to catch your cab to the airport. So on behalf of all of us, I thank you so much for the visit and all the work you've done.
Thank you so much, everybody.